All right, part one of our notes. You should already have those notes in front of you. Print them out. This is just to give you a little more background on the actual experiments. Okay, so this one, we're going to talk about the discovery of DNA, because so far, remember, we've talked about Mendel a lot and how he studied peas, and there was this idea that these traits had to be passed on somehow, but now we're going to talk about how we actually figured out DNA. The scientists that are responsible for giving us little pieces of information that helped Watson and Crick finally to put it all together in the double helix structure that you and I are very familiar with as DNA. Alright, so we're going to talk about Griffin, Avery, MacLeod, and McCarty, Hershey Chase, Watson Crick, and Franklin, and Chargaff. Okay, I would suggest you make a timeline. I wouldn't write down everything for each of these people. I would pick kind of their main contribution out and put that underneath the date. That way it'll help you keep it all straight. So first we're going to talk about Griffith. Griffith, he did an experiment on mice, poor little mice. He was trying to find a vaccine for pneumonia, which pneumonia can be very fatal and was even more fatal back then because they didn't have a vaccine yet. So in order to do this, he studied 2 different strains of pneumonia from mice, and what he did is he had 4 different groups that he injected with these different strains. So the first group, he injected the mice with the strain that caused pneumonia, which ended up killing the poor little mice. Okay, the second group, what he did is he injected them with a strain that did not cause the virus, so the little mice were just fine. The third group, he injected with the strain that causes pneumonia. However, he cooked the pneumonia first, or he added heat to it, which denatured its enzymes, its proteins, so that the mice were happily just fine. And then the last group, he took that strain that causes pneumonia, cooked it, and then he mixed it with some of the strain that does not cause pneumonia. Okay, and he was expecting, since he'd cooked the lasagna, and he'd already seen here that if he'd cooked the lasagna, the mice were fine, and the strain that does not cause pneumonia, he was expecting for those mice to be just fine, but what he found was that they died. Okay, this was really surprising. Okay, and so he came up with this idea that, obviously something from the cooked dead strain of pneumonia, something was being translated, or was being moved into the strain, the healthy live strain, the strain that should have kept those mice alive. So this non-pneumonia kind of strain was being transformed into the pneumonia kind, and that's where this idea of transformation is, that this there's some kind of information that can be kind of passed on and transform other DNAs. Okay, Griffith, by the way, was viewed very skeptically because everyone thought that proteins were responsible for heredity, and so the thought that, proteins, if you cook them, denature them, then the protein should have been dead, you should have seen happy, alive mice. Okay, Griffith said, no, there's something else, because I cooked those proteins, and those proteins were totally denatured. Okay, he said there's got to be something else that's transforming my happy strain. Okay, so the thought was that proteins were responsible, and people also didn't give Griffith much credit because everyone thought that bacteria genes were composed of different things than human genes. Okay, because they are much less complex than us. Okay, then we're going to talk about Avery, MacLeod, and McCarty. Avery, a lot of time, gets all the credit. Okay, this is 1944. But what they did, they worked together, and they basically used a process of elimination to determine what molecule is getting transferred. So they took Griffin's experiment and they expounded upon it. Okay, Griffin said it can't be the proteins. There's something else that's transforming the normal strain of pneumonia into the strain that kills. Okay, Avery, McLeod, and McCarty said, okay, well, let's start going through and eliminating these different things and figure out what it is. So what they did is they basically added a chemical that removed certain compounds like proteins. And so on their third try, they removed DNA. Okay, don't worry about how they did it. They added this chemical that removed the DNA. And they found that when they added that compound that removed the DNA, that there were no more lethal strands in pneumonia. Therefore, it had to be the DNA that was passing that information. Okay, so there you go. DNA was a molecule being transferred from one bacteria to another in Griffin's experiment. Sorry. Okay, now we're going to talk about Hershey and Chase. Hershey and Chase, they, in 1952, they studied bacteriophages. Bacteriophages, they are bacteria that kill viruses. They're very simple. Bacteriophages are basically, here, let me get all that up. Bacteriophages are basically DNA. Oh, apparently my pen's not working. Their DNA is surrounded by some kind of protein capsule. So if you're trying to figure out between DNA and proteins what's responsible for passing information on, that's like the dream subject to study, because you've only got two things you got to take a look at. Okay, so what they did is they attached radioactive markers to proteins and to DNAs. Okay, to protein, let's see, they added sulfur, and for DNA they added phosphorus. Okay, because we'd learn about structures of DNA, that DNA's got phosphate groups in there. Okay. Anyways, what they did is they did this big, long, complicated experiment where they tagged basically the proteins and the DNAs, and then they broke up the proteins and basically introduced it to E. coli. Okay, they broke up, the bacteriophages and introduced it to E. coli, and they found that the radioactive stuff that was entering the E. coli was not the protein, but it was the DNA. So they determined that DNA, not proteins, function as the genetic material. Okay, so here's a bacteriophage. You can see its head up here. Maybe, if I can get the pin to work. You can see its head. Nope. Okay, still not going to work. Anyways, moving on. You can see it here pumping out the DNA into the bacteria cell, which then that DNA goes in and it inserts itself into the bacteria's DNA, and then the bacteria starts reproducing all of the bacteriophage DNA, which then makes a lot more bacteriophages and kills the cell. Okay, so now we're going to talk about Chargaff. Okay, Chargaff, 1950, he was an American biochemist. By that time, it was generally accepted that DNA was a polymer of nucleotides. Okay, and it was also generally accepted that there were these four compounds: adenine, thymine, guanine, and cytosine. Okay, however, didn't really know anything more than that. So what he did is he did an experiment where he basically took a look at the amounts of adenine, thymine, guanine, and cytosine in species. He came up with these two important rules. They're called Chargaff's rules. Okay, first one is that species have different amounts of adenine, thymine, guanine, and cytosine. For example, sea urchins have 32.8% adenine, and humans, we have 30% adenine. Okay, each species is going to have a specific amount of each of these different bases. Okay, then he also realized that the percentages of adenine were almost equal to the percentages of thymine, and same thing for guanine and cytosine. Okay, so that led him to these pairing rules that you should know, that adenine always binds to thymine, guanine always binds to cytosine. Okay, didn't really understand why until we get to the double helix. Just Chargaff understood, okay, these two numbers are almost the same, like, I don't have the actuals written down, okay, but they're pretty darn close, and these two are pretty darn close. And so these two, therefore, have to be binding to each other, because if C was binding to A, then those numbers would be similar. Okay, so those are Chargaff's rules. Species have different amounts of adenine, guanine, or adenine, thymine, guanine, cytosine, and that adenine, cytosine, guanine, and thymine figured out A always bonds with T, and G always bonds with C. Alright, so now we need to talk about Wilkins and Franklin, and there's a lot of controversy about these two. Okay, well, not about these two, about Franklin and Watson and Crick. Okay, so early 1950s, Maurice Wilkins and Rosalind Franklin were using very sophisticated technology at that time. They basically were taking pictures using X-ray crystallographers. Okay, it's not an actual picture of it, it's like the reflection of the X-rays, what the X-rays are bouncing back. But what they were able to find from these pictures for the very first time, okay, they're able to see that DNA may be a twisted structure, maybe a helix, not quite sure. Okay, maybe two strands, and maybe nitrogen bases in the middle. Okay, this is actually really important. Here you can actually see, okay, this is a picture of Rosalind Franklin, and you can see this would be part of the helix here where it crosses over. You can see that they've got two strands in here. Okay, and this is really important because Watson and Crick, until they saw Franklin's photo, they actually, Rosalind Franklin, she also, she figured out that the nitrogen bases had to be in the center, phosphate and the sugars had to be on the outside because of just basically sizing and the way that it works and how it fits. That is really important because Watson and Crick, Watson and Crick, before they saw that, they actually had it backwards. So they had the phosphates and the sugars on the inside and the nitrogen bases on the outside. Okay, so James Watson is an American biologist. Francis Crick was a British physicist. Okay, in 1953, they basically took all these clues from Chargaff, from Franklin, from Griffin, all these scientists. They put all these clues together, and they built a wire tin model of what we now know as the double helix. Watson and Crick are credited with the discovery of DNA in 1962. Wilkins was also credited. Now, the controversy comes from the crucial part that Rosalind Franklin played in figuring out the double helix and that the nitrogen bases and everything went on the center, and yet she got no credit. Just kind of shows you a sign of the times. Okay, that is it for part 1 of your notes, and we'll see you back for part 2.